Okay, so ignore the name. My name's Peter. That's just how you write it. And it's quite a cryptic title, that one, maybe. So to expand it a little, I am going to simulate neurogenesis on massively parallel neuromorphic hardware, which is SpiNNaker. But the main research question that I have is what computational benefits can we see from neurogenesis, and how can we apply them in the context of SpiNNaker? So first of all, what is neurogenesis? Da, da, da. So, of course, I forgot to mention, I'm focusing on post-developmental neurogenesis, so in adults. And this only happens in the olfactory bulb and in the hippocampus. It also happens in non-neurogenic regions during lesions, but we ignore that for now. And it's exhibited in a wide variety of species. It's not just humans. Hey, look, a cute mouse. Why is this interesting from a computational point of view? Well, it aids in retrieving information which is very similar. So if you have two concepts that are quite similar to each other, the fact that you have neurogenesis improves information recall. It helps in storage of new information, provided that you can manage the addition of new excitatory neurons and so you have some homeostasis mechanism, and provides a longer term type of learning, which we don't currently have. And it's a different type of learning as well, for the fact that new neurons are more plastic than older adult neurons. But for the timescales, it compares to STDP. STDP operates on the timescale of seconds to minutes, while neurogenesis operates on the timescale of days, weeks, months. So that might be interesting. So what is SpiNNaker? You know already. So I pose a question to you. What can you do with the order of 100 million neurons and 10,000 synapses for each? Can you think of a model to fill that? Okay? No? Okay. Not for now. So what I'm interested with, what is my interest in SpiNNaker, is the fact that you can run, in real time, you can operate your learning rules, online learning rules, so you can see it learn stuff. But a limitation that I see is its topology, for the fact that the neurons and population are placed on the surface of this donut. Right, so you don't have any spatial information as you would voxel information in neural imaging or anything else neuroscience. So that's a limitation that we currently have. And this topology can't actually change. You can change the weights of the connections, but you can't actually remove connections or add nodes or add new connections. So the first step towards neurogenesis is synaptic rewiring or synaptogenesis. And I am trying to do this using a model of topographic map formations. So I start with a already formed network, like the one on the left, which was, trust me, formed by a chemoaffinity mechanism. And using an activity-dependent process of trimming and adding new synapses, we will get this kind of refinement in the weights. Why is this helpful? Well, I'm pretty sure we have the speaker on the left is somewhere in the audience. On the left, there was a study on mice. I think it was mice, not rats. And this shows in biology the structural plasticity that happens after rehabilitation from a stroke. Right, so new synapses can form. And on the right, there is a computational model of that, where you can see concepts, associations between concepts being regained after artificially inserting a lesion. So we don't currently have that on SpiNNaker. Cores can die, but you can't really recover from that. So what am I doing on SpiNNaker? As I said at the beginning, I'm not telling you. Come and ask. So we can talk about memory and homeostasis and interactions with other learning rules. But for now, more enthusiasm. What can we use this for? We believe that we can use it to play breakout. So you have a state space that represents everything regarding breakout, where the ball is, where the paddle is. So we think that neurogenesis will help with this by increasing the state space, helps in storage, and for the fact that it's a longer term type of learning, then you can have transferable knowledge. So you can take this and say, we can play Pong now, quite easily, maybe. Some other plans and ideas that we have: actually add or remove neurons, so neurogenesis. And devise a homeostasis mechanism for all of this. And potentially, which would be a very tricky thing indeed, modify router entries on the fly. If you don't know what that means, it's okay. Come and ask me. Also, for the fact that we don't actually have spatial information, it would be a good idea to select interest volumes or voxels on the host and then place them on the 2D thing that is the board and the placement of the chips. And rule-based developmental model, sort of something like L systems or a grammar to define how a population can grow from 1 neuron to 2 to 10, and then fill the whole machine, 100 million neurons. But there's time. It's only 21,000 hours if I don't sleep or eat or take a break. Totally doable. But regarding the key question that I had, watch this space, it's not done yet. I'm sure there's lots we can load and lots we can implement. Thank you.